Hi everybody, good afternoon. I am very happy to have Benjamin Scholl here, our speaker today. So Ben did his PhD at UT Austin with Nicholas Priebe, and his postdoc with David Fitzpatrick at Max Planck Institute, Florida. And since last year, right, he started his lab at UPenn, and he's going to tell us about whatever he did in here. Have fun.
All right, thanks, Rob. Cool, yeah, it's awesome to come visit. It's the first time I've been to Hebrew University and come to ELSC. I've actually know many people here by name, but not by in person. So it's a lot of fun to actually put the faces to the names. It's actually it's been almost 10 years since I visited Israel. The last time I was in Israel was to see Yossi, and he made me patch in a course for 3 weeks. Though I spent a lot of that time swimming in the Red Sea. So, and I'm really happy to be back and see all the interesting neuroscience in Israel. So today I'm going to talk primarily about one of the last projects that I worked on as a postdoc, and then some stuff that I've done more recently, and newer things that are a continuation of some of that work. And actually, some of it I haven't. This will be the first time I've talked about it, so I'll be curious to hear any interesting questions or thoughts. The primary interest of mine and many of my recordings and a lot of the work that I've been doing is not entirely from this perspective, which is, I would say, more of a conventional or traditional perspective in modern neuroscience right now. And that is like analyzing and observing and studying the populations of neurons at the cellular level, the cellular perspective. So this is two-photon imaging, the population of cells. This happens to be from the ferret visual cortex. And everyone here, I'm sure, can appreciate the activity of these cells, and there's a lot of dynamics and there's a lot of things that we could study about these neurons in the context of sensory representations or behaviors and such. But it's really important to remember that each one of these neurons has a complicated nature itself. Each one of them has dendritic arbors that extend far out into the cortical space, and sometimes up to 500 microns or a millimeter. And along those dendrites that extend in 3D, there are hundreds, if not thousands, of synaptic inputs that are impinging on that neuron. So each neuron itself actually has a population, which itself has dynamics within the neuron of all the different inputs that are impinging on it and driving it. And the synaptic populations, I believe, really underlie the computations and define the computations that cells do and the operations that they perform. So I'm very, very interested in understanding. Oh, nice. And being able to measure and dissect and understand the synaptic populations within neurons and how they drive them. So when in David's lab, I started this work, and we use the visual system. And the visual system affords a lot of advantages, and mainly that input-output transformations between neurons can be very clearly and classically defined. Mainly, I think many of you probably have seen something like this on the right, which is just the canonical tuning curve. The y-axis is the somatic response, could be the action potentials, could be the memory potential. And the y-axis would be a number of bars of different orientations that are presented to the animal. And so this is the canonical orientation tuning curve that we might measure from the soma of these cells. And so the first question that we asked, almost 10 years ago now, was what are the inputs that drive the cell that would underlie the selectivity that we see in the output of the cell? Is there a simple understanding of what those inputs might look like? And there was the main idea at the time, which I think now has really been hammered down to and known to not really be the case. But 10 years ago, and more so, there was really one idea of how you would construct this, how you would wire up a cell in order to generate this kind of tuning. And that was that there was some form of functional specificity. So basically the inputs that the cell receives that drive these responses at the output in the soma really share the same properties. So the cell has a bunch of inputs that are very similar in tuning, and those together really just drive the activity of the cell and define its operation. And there was a fair amount of evidence for this. and older measurements of connectivity, and of course things like looking at an evidence in support of like-to-like connectivity itself might support this kind of idea. But alternatively, it could be the case that something else is more interesting or more complicated is happening. Something that, there's a diversity of different kinds of inputs that the cell receives, and the tuning that you see at the output of the cell or in the soma is derived from something which I term functional amplification, which just means that a subset of these inputs within the neuron probably have a greater influence on the soma itself and would shape the selectivity that you see in the output of that neuron. Okay, so the very, very first question that we had those years ago was, do the synaptic populations within neurons exhibit functional specificity or a diversity? And the primary method in which we study this is two-photon imaging of single cells, and specifically the dendritic spines on excitatory neurons. And so calcium imaging is pretty much the best way that we still have as neuroscientists to be able to assess synaptic inputs onto cells. And dendritic spines are a thing to study because they are the site of excitatory inputs onto pyramidal cells. And we use the ferret model mainly because it's a very good model for vision and development and a number of other things. The way that this technique really works is a combination of both the spatial, the point spread function of two-photon imaging of allowing you to see very small objects, but also the sparsification of the expression. This is absolutely critical. And so the way that we do this is that we just drive Cre and we dilute it, so only a few neurons are expressing the Cre virus, and then we express a Cre-dependent construct such as GCamp, and we can use anything else, obviously, and all the cells. And thus we end up with, depending on the dilution of your Cre and your particular virus and promoter, you try to titrate down to have just a few cells where you can see individual cells, their dendrites, and the individual spines. And then you can zoom in on individual segments, where you can very nicely resolve dendritic spines and their individual signals. So this is an image of some data that's being collected. Up here in the top left corner is the visual stimulus that we're presenting to the animal, so different orientations, just like that I showed you before. These bulb-like structures are the dendritic spines on the branch, and you can see there's kind of a variety of different signals here. Some of the signals seem to be very isolated and just independently in the spine, and so we will call those synaptically driven calcium signals in these spines. But occasionally you also see a global event that occurs across the branch, and those are due to back propagating action potentials that are going back. Yeah.
So which synaptic context do you see? Do you see NMDA? I mean, is this like a specific to NMDA?
Yeah, so based on slice physiology that was done by Carl Svoboda previously, the majority of these signals seem to be driven by NMDA and voltage-gated calcium channels that are activated. So presumably there are smaller inputs, weaker inputs that are AMPA only, and we would not be able to resolve those. Yeah. So we're always biased in that sense with the calcium. Although I'll show you later that we do see small inputs, and there are many reasons why that we actually can resolve them with calcium. So the sort of typical way to analyze these kinds of data is to look at the calcium responses in the soma to these different stimuli. You can see the soma prefers this particular bar. And here's a bunch of spines on this dendrite, and they have a bunch of different responses to the bars. And you can already see that there's a variety of different kinds of responses, a variety of different tuning within the inputs. And there always appears to be some diversity. And of course, we can sort of look at the representation across the entire cell. So all the different dendritic segments that we were imaging, we can go and we can look at the tuning of the individual inputs that is denoted by this circle, the color being the orientation preference of that individual input. So the soma here is in green in the center, and these are all the different synaptic inputs. And very clearly from this data, this was very surprising to us, especially in an animal which has a columnar organization of orientation preference. We did not expect to see such a diversity of orientation preference inputs throughout the entire cell. But this image, this diversity, actually turned out to be a pretty common theme throughout all the work that I did as a postdoc. And so this was one of the first studies that I worked on in which we looked at a fundamental thing, just orientation preference in ferrets. But when we looked at other features such as the representation of visual space, visiotopic space, so as shown here, surrounded by the receptive field of that neuron, so relative to the receptive field of this cell, you see there's a wide representation of visual space represented across the dendritic arbors. The same thing for motion in terms of the preference of the direction of visual motion being in the preferred or opposite direction of the cell, and even in terms of eye preference or ocular dominance. So here we're looking at whether inputs are responding to the left eye, the contra, or the right eye, the ipsi eye, or both. Diversity seems to be a common principle. And actually what's been really interesting and confirming about this is all work that was done in the ferret, but now there's studies that have been done in the mouse, both in the awake mouse and both in sensory areas and more decision-making areas like ALM and parietal. And in all these experiments that have been done, everybody has basically measured something similar, seeing functional diversity of inputs onto neurons. And even actually in the primate, now there's been a study in the macaque, in V1, looking at individual cells, and they see something very similar to what we see here. So this seems to be really a common principle. So given this functional diversity, how do you manage to get something selective at the output? That's the natural question. So obviously, this is kind of just a one view of the data that you can look at. You could average all the responses from all the spines. You can get this sort of prototypical sort of predictive sub-threshold tuning curve and compare that to the output of the cell. And you could always try to pass it through a threshold, and that might do some work in terms of sharpening it, but honestly, it won't take you all the way. And this bump, the fact that you can capture the preference of the soma is seen in the data in the sense that there's always a bit of a bias, and that'll come up later too. But I was very interested in other ways in which the arrangement and the particular properties of the synaptic inputs might be able to enhance specific ones or amplify their influence and perhaps get you closer to something to more like the soma. And so things including the local organization of inputs. So if inputs similar properties are nearby each other and they're coactive, they could take advantage of dendritic nonlinearities, and those could enhance their effectiveness in terms of driving the output. Very simply, there could just be a weight distribution, a functional weight distribution. So if inputs that are co-tuned to the output are just bigger and they have more a larger weight, they could have a greater influence themselves. And of course, this is all in terms of excitatory inputs, looking at dendritic spines. And of course, inhibition is always is a major player in these cases too. And so inhibition could, of course, cleave away certain excitation or selectively enhance particular sources of input. And these are 3 different studies that I did actually as a postdoc, but I'm only going to focus on, and I can tell you that there is clustering and inhibition does have a role, but today I'm just going to really talk about the synaptic weights. So looking for a functional bias in the synaptic weight distribution. So there was a hypothesis that already existed in the literature from previous studies, and that hypothesis was that the output of the cell is really driven by a few strong co-tuned synaptic inputs, and this is how you would construct selective output in your cell. And there was a fair amount of evidence, but there's 2 very specific studies that I feel really tried to hammer this home. So, and one of them is from Tom Mercer-Flogel's lab, and in here they did this incredibly heroic experiment where they did two-photon imaging of individual cells to map their sensory response properties, and then they sliced the brain to find the exact same cells in vitro, and did intracellular recordings, paired recordings to record the connections between neurons in terms of the EPSPs. And what they found is effectively, if you see here on the right, that cells with more similar responses have larger EPSPs that are more likely to be connected, and those connections are more likely to be larger. And I like what they wrote in the paper. Each neuron receives the majority of its local excitation from a small number of strong inputs provided by the few neurons with similar response properties. Okay. Now, the thing is, this paper is not looking at individual synapses. They're doing paired recordings of nearby neurons and recording EPSP. So they don't have a direct measure of the exact functional response property of individual inputs. So the question was, how do we actually, we have ability to measure the functional response property of individual spines, but how do we assess a metric of strength to those inputs? So what's also well known is that ultrastructural morphology of synapses is a good metric of synaptic strength. So the release probability of a synapse is highly correlated with its size. And there are many, many studies showing this, that basically the size of the spine head is correlated with the number of postsynaptic receptors that are there, as that's correlated with the size of the presynaptic glutamate, which is correlated with the size of the presynaptic vesicle pool. So all these things are in line. And also, but actually, there was an interesting issue with this, which was that it wasn't actually totally clear whether this ultrastructural element would be correlated to things you can measure in a slice, like the EPSP amplitude. And actually, around the same time that my paper came out, another paper came out from Kevin Martin and a few other groups doing exactly this experiment, where they actually did paired recordings between two neurons, measured the EPSP that would be driven from one into the other, and then did EM to find the synapses, and then look at the size of the PSE area of those synapses. And here they show that there's a nice relationship between EPSP amplitude and the postsynaptic density of those synapses that presumably drove that EPSP. And so it really seems that the ultrastructural properties of the spine will give a nice measure of strength of the synapse. So what we had to do, though, was be able to find a way to combine these two modalities. So we have in vivo two-photon imaging, but we need to now be able to measure the ultrastructural properties of the identified and visually responsive spines. And so we came up with a method, more or less just a pipeline and combining materials and methods that existed previously, where we did in vivo two-photon imaging, and then we recovered the slice, we fixed the brain, we recovered the slice that we're imaging, finding the same cells, and then once we recovered the cell that we had been previously imaging, we prepared that chunk of tissue for serial block phase scanning electron microscopy, and which it was centered around a particular cell. So then of the cells that we mapped, we were able to, once we could find them, we then took them to the EM and did volumetric EM imaging of that block. So the main trick to this procedure had to do with just like identifying features that would allow us to correlate between the fluorescence images and the EM. So you'd start with a piece of tissue, you'd see your cell there, you trim it down, but basically once you stain the tissue and you prepare it for EM, which is basically impregnating it with a ton of heavy metals, you would lose your fluorescence. You no longer see where your cell is. And this is the pin that the tissue would sit on for the serial block phase microscopy. But if you do a low resolution scan of this block, you can very clearly see a lot of different features, including blood vessels and cells. And so we were able to devise an algorithm that would just very easily correlate these features with a fluorescence stack, from a confocal. And just with sectioning a little bit, we'd be able to predict exactly the X, Y, and almost the Z location of the cell that we were interested in imaging. And I should say that in this in the first variation of this study and this work, we decided to identify the soma of the cell because we knew that would be very big, and we would then reconstruct out. But since then, they've actually moved this technique a lot forward, so they now can identify individual dendrites, and they no longer have to reconstruct from the soma out. And so it made some progress there. This is just an example image of what one of these sections can look like. I like looking at EM images a lot now. I didn't think that would be the case, but we learn new things all the time. And so here you can see a synapse. This is the postsynaptic side. There's the spine. This is the postsynaptic apparatus. There's the PSD here. And here's a number of vesicles on the presynaptic side. Now this is serial EM. It's not going to be as high quality as TEM, but there's advantages for this strategy. And one thing that I do really appreciate about the work that was put into making this project happen is that these are really nice SEM, and this is an adult ferret, which is not, if you do any EM, there's a lot of work you need to do to optimize the perfusion solution and the exact preparation so you get really nice contrast, which we're able to do in this case. And that's a lot harder with animals that have larger, bigger, and more dense brains. So here's an example cell in which we were able to reconstruct the soma and out into the basal dendrites. Here's the cell from a low magnification projection imaged in vivo under the two-photon. Here's zoomed into the soma. Here's the same cell. Then here's a branch in the two-photon and from the EM reconstruction. So these are all the volumetric reconstructions, and you can see the spines here, the same spines there. And actually, once you have everything kind of aligned in the same geometry, almost everything pops out. The main thing that is very interesting about this is that, obviously, not surprisingly, you recover about 30 to 40% more spines than you saw in vivo. So the EM shows a lot more small spines and things that weren't resolvable before. So synaptic strength is diverse. This is well known, but it was interesting to see within our own data set that here are two different synapses, and they could actually differ by 1 or 2 orders of magnitude. So the spine head volume here is over a micron cube. This is the bouton. This is the snaking PSD. This is the spine head. And here's another more simple spine, much smaller. One of the coolest parts, though, about this data set is that we were able to then assess the number of presynaptic boutons per spine. And this was a little surprising to me, but basically almost all of our data was a one-to-one connection. So almost every single spine, is like 98% of spines received a single presynaptic bouton. This is not the case across all brain areas. In some areas, like in the hippocampus, you might find that there's multiple contacts, multiple boutons innervating the same spine. But here in the basal dendrites of the ferret, it's one-to-one. And this is a nice confirmation of the functional measurements we made, so that we were a little bit more, we have a little more support towards our functional measurements in the calcium. So those are reflections of single presynaptic neurons rather than multiple inputs converging at the same time. Okay, so back to the question at hand. If there was a functional bias in synaptic strength, then this would be the relationship that we would expect. So the y-axis here is the spine volume of each dendritic spine that we imaged and then captured the EM. This is the PSD area. The x-axis here is the similarity between spine and soma in terms of orientation preference. So 0 would mean that they have the same tuning, and 90 degrees would mean they're orthogonal. And if there was a functional bias, as suggested, then you would see that all the inputs more closely to 0 would be much larger. But in fact, we didn't find this. So we found that both strong and weak synapses were equally likely to be similar or dissimilar to the soma. So the inputs are all over the place. Now, this could be explained by the fact that spines themselves have a complex geometry, right? So we're just comparing the spine head and the PSD, but we're ignoring things like the neck. We can provide filtering, and in some cases, the necks of these spines can be up to 4 microns long. So to try to assess this possibility, I turned to some modeling, and so we took the entire geometry of the spine and we translated that into electrotonic properties, put it into a biophysical model, and then simulated what the inputs, what the effective somatic voltage might be from simulated inputs onto these spines. And so when we try to do this and take account for all the geometry, we still see that there's no functional bias. So but basically, even in the simulated case, co-tuned and orthogonally tuned spines are equally likely to be weak or strong. Okay. So synaptic strength distributions alone don't seem to predict somatic tuning. This is an example. This is the average of all the 5 cells that we have. So there's this nice tight tuning here. And I was curious as to whether we maybe get a different view from this if we look at the data in a slightly different way. So what we can do is we can go and say, well, what is the strength distribution of spines recruited for this stimulus versus this stimulus? So for the stimulus that drives the somatic output versus one that doesn't drive the somatic, so it's orthogonal. And we can just go and say, let's take all the inputs that showed a response, so they're active. And there are many ways that you can do this. This is a very simple way of doing it, of just saying, like, you have to have a reliability of greater than 50%. And then you can see the distribution of the weight distribution from these synapses within these cells. And here is plotted the mean, the median, and the interquartile range of that distribution. And if we do this for all these different stimuli, you see something that's basically more or less flat and has some noise to it, obviously. Now, I have to say, like back when I was doing these experiments and analyzing these data, by the time I got to this point, I was a little bit frustrated because I hadn't really seen anything that would explain what was going on. After a long conversation with my postdoc advisor, David, somehow it came to the idea that we should just look at the numbers. And when you look at the numbers of inputs that are active on any given trial, there you begin to see this beautiful tuning that you see before. So this is the number of spines that contribute to each one of these distributions. And you see that the number really is showing the same kind of tuning structure. And of course, if you incorporate also the weights of those individual inputs, you can recover something that looks a lot more like the somatic tuning itself. So it seems like we're seeing strength in the numbers of inputs rather than a functional bias. Now, there's a little bit more to the story that I quite like, actually, and that's something that I mentioned, but in another study where we saw that one way that that inputs can be effectively stronger is if they're co-active with other inputs nearby. So if there's clusters of inputs that are co-active, that this could enhance their effective contribution in the cell. And so this is a study in which we saw that sensory drive could identify spines with the same receptive field properties, the same sensory response properties that were very tightly clustered in the same region, and we could quantify this and see that there was these short, small clusters of spines that were highly coactive within about 5 microns, sometimes 10 microns. And so we were wondering, well, can we look at this data in a different way in terms of the strengths of these particular inputs? Is it more likely that weak or strong inputs might be clustered? And so this is the EM data set. So this is a similar a plot of just the EM data that we have. So the y-axis is the pairwise correlation between inputs. The x-axis here is the dendritic distance between those inputs. And you can see that there's far more correlation for inputs that are very close to each other than far away. The dashed line here is shuffle corrected. And so we can play the same game by just looking at the strong or the weak inputs. And what is really surprising is that basically if you look at only the strong synapses, the biggest synapses, this relationship goes away. And for the weaker synapses, you still see this relationship, and maybe it's even a little bit stronger. So this would suggest that the weak synapses are most likely to be clustered and coactively clustered together. And what's really interesting about this is that synaptic strength distributions in cortical tissue in these cells exhibits a log normal distribution, basically meaning that there's a lot more small synapses than there are big synapses. And so it's not just that, and so when you put these two things together, that there's potentially strength in numbers and that the weak synapses are more correlated, they also exist in greater number overall. So one thing I kind of like about this data set is that it sort of suggests that instead of the strong, big synapses that are co-tuned being the major drivers of selectivity and the somatic output of the cell, that in fact the weaker synapses that are more numerous and clustered may have an outweighed role that's underappreciated. So that's so that's about what I was saying. But okay, so one thing that kept coming up over and over again in discussing these data is there any structure-function relationship at all? And we didn't see it in terms of the co-tuning between inputs and outputs, but is there any other response property or activity in the spines that we could see that is related to the size or the strength of the synapses? And there was one feature that was wildly significant, and that was the tuning selectivity of the individual inputs. So here I'm just plotting the tuning selectivity. It can be orientation selectivity or direction selectivity. The y-axis here is the spine volume in blue, the PSD area, and then the simulated one. Over here are just two examples of what a highly selective input would look like compared to a more broadly tuned input. So basically, when this is vector strength index, so a 0 would mean a completely untuned input, and a 1 would mean perfectly tuned. And in all these cases, basically, we see that inputs that are bigger tend to be the ones that have the tightest tuning. And honestly, other than seeing this data, I didn't really have much more to say about it, and I didn't really know where to take it. But a friend of mine, Claudia Klopath, actually saw this and became highly interested in started a project that I was very lucky enough to be a collaborator on in exploring how this phenomena might relate to the overall findings we're seeing, and how there could be strength in numbers, and how you might construct that within an individual neuron. She and her grad student actually developed a little model using a learning rule to study how these cells might be constructed, and how we might observe these how she might observe these exact same phenomena that we saw in vivo. So the learning rule they called the presynaptic variance rule. And basically what they did is they constructed a model where there's a postsynaptic neuron receiving a bunch of inputs, and those inputs have a variety of different tuning. They can be wide, they can be narrow. There's some inhibition, of course, but they're mainly just the plasticity is going to occur in the excitatory inputs. And then the learning rule just says that basically there's some learning rate, and it's going to be multiplied against the response, the activity in the presynaptics minus some background activity, and there's some decay rate term too as well. And so when they do this, they let it run and then stabilize, and they get they can get a weight distribution that looks very similar, that has at least the same shape of things that we see. There's this basically long-tailed distribution, a lot more weak and small things. And very interestingly, what they also see is that the largest weights have the highest selectivity, just like we saw in our data. So inputs that are the most selective get assigned basically the largest weight in the postsynaptic neuron. And this model actually generates neurons that have tuning that look more or less like canonical orientation tuning. So they let this thing convolve and run and get the weights mature, let the weights mature. And these are a bunch of examples where they get this nice orientation tuning. And just like in our data, they see that the synaptic weights that they end up with are not correlated with the preferred orientation, but they still see this nice relationship in terms of selectivity, which is quite nice. And then they finally turn to in the model, they turn to the same question about strength in numbers. So then they look, and these neurons, they can look at the distribution of the inputs, just like I can look at the distribution of inputs in vivo. And they see that basically the probability distribution shows a bias, and then here's the tuning of the total input, and that bias translates into something similar to what we see in vivo, and that basically the explanation is the same, that the number of active inputs that are contributing to a particular stimulus on a given trial is the greatest indicator of and drive of the selectivity of that neuron. So this little model that I've been working on, that they've been working on, that I've just been collaborating with, sort of suggests that potentially a plasticity mechanism that is based on the presynaptic input alone might actually be responsible in terms of shaping the size of inputs and help define the output, the properties of the output of the cell. And this is sort of it's sort of anti-classical in the STDP Hebbian framework in terms of spike timing, pre-post correlation and spike timing dependent plasticity. But it's actually not that crazy of an idea. There's a lot of different experiments that sort of rely on this principle all the time. So there's a lot of evidence in just thinking about looking at uncaging done on slices. So this is an example of glutamate uncaging on an individual spine. And this is essentially basically having lots of correlated activity on the spine by a glutamate uncaging, and this induces LTP. So we've known for a long time that without even sort of invoking a spike from the cell, there are methods in which you can induce plasticity of just independently of an individual spine. So and this is now I'm moving forward a little bit in terms of this work to sort of explore developmentally whether local and how local and global mechanisms shape the population of inputs within neurons with visual experience and such. Okay. So in the last 10 minutes or so here, I just wanted to talk about a little bit of new data that we have from this same data set. And one of the cool things about EM is that there's a huge amount of information you get from these data sets. And it was always sort of a bummer to me that we would do all this work and we'd make this one specific measurement to correlate with the functional the activity that we measured in these spines, and then it would be over. But there are a number of features that we can see, such as apparatus and astrocytes, and in particular the mitochondria. And there was a student who was very interested in annotating mitochondria and looking at where they are located in cells. And so that got me thinking about, well, I wonder if there is any particular organization to mitochondria within cells. And this is all postsynaptic, by the way. So, mitochondria are thought to provide a metabolic support for neurons, and there's a lot of study on mitochondria in the presynaptic side, and a lot is known about their role in terms of regulating the glutons and activity and strength, and not as much on the postsynaptic side. I would say that that part of the field. There's now are people beginning to study their organization and the anatomy of these things. So I was really just curious about whether there is a postsynaptic organization of mitochondria in the dendrites and in the spines, and then specifically whether that particular organization actually showed any interesting relationships to the properties of those spines, because we have this ability now with these data to be able to compare structure and function. So I mentioned this, but basically from the postsynaptic side, there's 3 different cases that we would see in the data. So in one case, you can see a spine with an active PSD, and there's no mitochondria either in the spine head or in the dendrite. And more often, there's a mitochondria nearby that's within the dendrite itself. And then occasionally, you actually see a mitochondria inside the spine head in itself. But this is actually the minority of cases are here. So to analyze this data was a little tricky in the beginning because there wasn't a lot of studies out there of examining these 3D structures with respect to a single spine. And honestly, we started by trying to measure distances and compare along track lengths and things like that, but realized that this wasn't a good way to do this because these are 3D structures, and so we really need to take into account that aspect of that nature. And so what we ended up choosing to do is sort of do this volumetric analysis to ascertain the fraction of surface area within the mitochondria in the dendrites nearby the spine. So this is an example of a two-photon image, and here is the reconstruction in the EM. These are different colors here, other than the purple, are individual spines that were visually responsive, and the purple here are the mitochondria within this little segment of dendrite. And so what we did is for every single spine, we decided, due to just some time limitations, we decided to do 2 different distances. So we took a 1 micron radius around that spine and computed the total area of mitochondria with around that, and then also did a larger one at 5 microns. So we have these 2 measurements. We also did 10 too, but basically we wanted multiple measurements along the dendrite to get a sense for whether there's local versus global organization of the individual inputs. And so this is actually really surprising. I had sort of this kind of baked hypothesis in my head about what I might expect, and what we saw in the very first analysis is not what I predicted at all. So this is what I showed you before. This is the tuning, difference between spine and soma. And so for every individual input, every individual spine, we looked at the difference in orientation preference just the same. So 0 means they're the same, and this is supposed to be radians, but. And this would be opposite above here. And so the x-axis here is the volume of mitochondrial membrane within 1 micron for around that individual spine. And what we saw is actually there's this very strong relationship where inputs that are dissimilarly differentially tuned or oppositely tuned with the soma have more mitochondria around them. So it's actually counter to what I had predicted in my head. And I'm not showing this here, but this relationship goes away if we look at the 5 micron radius or the 10 micron radius. So if you sort of examine the total fraction of mitochondrial membrane within the dendrite at larger scales, there's no more relationship. You only see this relationship for very local mitochondria around the spine. So differentially tuned synapses seem to have more mitochondrial support. Another thing that we looked at was the distribution of inputs within that dendrite. So we can take a spine and then we can ask how similar are the inputs of nearby spines. So they can either be more homogeneous, meaning that they all are co-tuned, or can they be less homogeneous, so there's going to be more diversity or heterogeneity. in terms of the orientation preferences. So lower numbers would mean more homogeneity, higher numbers would be more diversity. And here we saw that actually greater diversity was correlated with more mitochondria being closer to spines. So this again was a little bit surprising, but it sort of seems that mitochondria are positioning themselves to be supporting functionally diverse branches, or at least branches that have a diversity of orientation inputs. Now, these are only spines in which there was no mitochondria in the spine head. So we're only looking at mitochondria in the dendritic shaft. I didn't tell you anything about ones that are in spines, and they do see here. But these ones seem to have very different, very specific properties, and that are a little bit more in line with the literature from presynaptic Glutamate. So, and this kind of makes sense. So here's a reconstructed spine. This is the PSC. Here's the mitochondria. And basically, spines that have a mitochondria inside tend to be larger. They tend to have larger PSCs. And not surprisingly, based on the relationship that I showed you before, those that are larger also tend to have a higher selectivity. And now, I wish we were able to do more interesting analyses in terms of like looking at tuning correlation, similar to the soma and things like that. But unfortunately, the number of spines are only 12 out of almost 200 that we report, 6%. So we don't have the statistical power to really say anything more interesting. And I can't say whether mitochondria in spine heads serve a different property than ones in the dendrites. That might be the case. I don't really know, but we'd have to be able to collect more data. But this actually became a little bit of a fascination of mine of how would I collect more data if I wanted to do this, because there's a lot of different things that could explain this. I mean, one, for example, is that actually we're just getting this snapshot in time. We did this two-photon imaging, then we sacked the animal, and we did our EM. Well, maybe mitochondria are actually quite dynamic, and there's some ongoing plasticity, and these are actually moving around, and we've just sort of froze them in time. So what would be better to then go through this whole EM step again is be able to monitor mitochondria in vivo and watch them and make these similar kinds of measurements. And so that's what I've been doing in the last 2 months, actually, is I had to do two things. So first we had to develop a viral strategy to sparsely label a cell with two constructs robustly. And then we also had to tag mitochondria. So this work is actually done in collaboration with Tel Aviv, up in Tel Aviv University. So we made this construct that contains Cre-N flip, so we can transsectionally sort of express things independently in individual cells. So this is an example of using expressing both GCAMP and SciRFP in the same cell. It works extremely well. SciRFP is a very nice far-shifted red protein. And so what I asked Paul to do with this construct is basically add a mitotag to it, so we can do the same kind of procedure, and we have both GCAMP and the mitochondria labeled in the same cell. And so this is an example. This is not the best image, but this is an example that just came off the microscope a month ago. And so here's the soma, and it's chock full of mitochondria. And here are actually these are dendrites. You can't really see the structure in these dendrites, but you can see a little bit of the structure just due to the mitochondria. And each of those bright objects is actually a mitochondria. So this is just the beginning, but I'm hoping that we can iron out this approach and validate it, and then basically begin doing structure function imaging in vivo, tracking mitochondria postsynaptically, going in spines, and correlate it to their activity. And hopefully I'll do this all during development too, where we might see interesting dynamics. Okay, so if we're turning back to the original question, in terms of how do you build, selectivity at the soma, and in terms of the visual system, instead of specificity, we see that there's a diversity, and their forms of functional amplification seem to maybe involved in constructing that somatic selectivity. So I told you that synaptic inputs on the cortical neurons are diverse. Both strong and weak inputs are equally likely to be similar to the soma. And instead of a functional bias in synaptic strength, we see that the strength seems to arise from the number of synapses. We're starting to explore how a presynaptic variance rule might describe the emergence of this. And then finally, I showed you some new data that suggests that local mitochondria may actually support the functional diversity that we see and that has been seen in all these different studies. And with that, I'd like to thank the people in my lab, as well as the MPFI EM core, which none of this work would be and the Fitzpatrick lab, obviously, and a few of my collaborators here. As Yoav mentioned, I'm now at Penn, just getting started and doing things on my own, which is interesting, and thank you very much for your attention. Yeah.
Yeah, so my sense is that this is still sort of consistent with the linear neuron hypothesis, right? In the sense that if you have a linear neuron, you expect the output to be a weighted sum of active inputs. So if you have more inputs of a particular orientation, then you'd expect, if all the weights were the same, you'd expect the output of the neuron to be more the same. And I don't know if you did the analysis here, right? But if you just multiply each of the weights by the activity, that should be what you expect.
Yeah.
Right, so did you do that analysis? Because you have the activity and we have the weights.
Yeah, so it's a little tricky because we have the activity and we only measure, so there's one caveat to this, we only measure, one or two dendrites at a time. And so we have this sequential measurement. So some dendrites, and we're getting sort of a random sampling of inputs each time. So you don't actually have quite enough inputs, especially like inputs that are hundreds of microns away. I tried actually putting these things into a model because yes, you can. When I wanted to test the linear model, I wanted to test it given electrotonic passive filtering and stuff like that. And in this case, the problem is that it's not generally enough drive to really see something at the soma. And so then it became an issue of like, well, how would I construct this model from these inputs to really test this hypothesis? I think what you're asking is an excellent question, and I'm really interested in it. But I think that it's hard to say from this data one way or the other. Like obviously there's nothing in the data here that suggests that the clusters, for example, invoke nonlinearities, right? It's not like we have a really clear measurement of them. But I'm very interested in actually building a more comprehensive model to incorporate in vivo data to study this. And right now we're doing one of the issues with this, though, like I said, is that you need to simultaneously sample lots of inputs. And here we were really focused on just collecting good data. But now there's a lot. The technologies out right now are like much better to be able to do much larger sampling, where you can image like 5 or 6 dendrites simultaneously. And I think that this approach will allow us to collect the actual activity that will allow us to really more directly test this hypothesis. Yeah.
First of all, you mentioned early on, you showed the result from EPSP measurements. You said, well, this is multiple contacts or to do with the soma, but this is what the cell cares about. So the question is, are your results consistent with that?
Right. Are my results consistent with this?
That the EPSP is strength is selective.
Yeah. So actually, if you take those results, I actually don't think that the previous results and my results are in contrast. I think that they are in line, and I think that the difference is, as we talked about today, is the definition of strength. Is how do you define strength? So the terminology in those papers is actually like sometimes unitary strength. But I would say there's the effective strength, which is defined by these parameters of the size of the synapse, the location of the synapse, the number of contacts, the number of contacts.
Okay, but this is an alternative explanation for the selectivity.
Yeah.
It's an alternative explanation to you.
Yeah. And I think that, there's even this study, so this study...
So you don't need strength in numbers, you just need strength in EPSP.
Well, no, because I think that the EPSP size is also correlated with numbers.
But the numbers of contacts, they're the same source. I don't think that's what you have in mind when you say strength in numbers.
Yeah, yes. So I see what you're saying, yeah. There's evidence in both. So there's also evidence... So along with their evidence, which is more or less, for me, like this is a confirmation that there is that strength can contribute. That's the unitary size can contribute to EPSP. It's also known that the number of contacts can also contribute to EPSP. So this was shown by Bert Sockman and Dan Feldman, and actually in the and Clay Reed's EM data set, where they are looking at the somatic responses and saying, okay, which cells have the same tuning, when they don't look at the sizes of all the synapses, but they did identify all the potential contacts between neurons, and there they see the same thing. So they see that cells that are co-tuned have more connections between each other. They don't know what the sizes are, but they know that there's more.
I have another question about the model. So I'm confused if it is purely presynaptic and there is no competition on the postsynaptic in the model, so how do you get selectivity? What is different between neurons with different presynaptic neurons with different orientation selectivity to make selectivity in the numbers?
Yeah, I think in this case it has to do with small biases that just get amplified. But I mean, I don't think this is just the starting place. And I for sure don't think that. And my interest in this was to explore this further because obviously like spikes do occur, and there's many other things that are being ignored here. I think that it's an interesting idea to explore as to how these presynaptic mechanisms might contribute. But I don't think that they alone are the sole reason for deriving selectivity or strength. Yeah, Omar.
I was wondering, in the comparison between the EM data and your two-photon imaging, do you have actual numbers on what is the number of spines that you see being active, and what is the number or the percentage of the large spines specifically that you see being active?
Yeah. In terms of just the imaging data, or
like between EM, what is the total number of spines on a neuron?
Yeah.
And in two-photon, what is the actual number that you see being active?
Yeah, I mean, I try not to go down this road because, and the reason is because there are many more. So two-photon it has caveats, right? And so one of the biggest caveats is that you only see things that are resolvable via the point spread function. So we miss a lot of spines. And so the total number is always much, much larger than we could even say from the two-photon. And it's the case that there is a bias towards larger spines. We do see small ones, but there is a bias. We quantified this very carefully in the paper. We were very honest about it. But basically, our method of looking at activity is very much biased towards larger ones. So, I mean, and presumably probably not seeing a lot of the ones that are fully amplified. And also we're missing ones that are on the shaft too. So we're really just seeing like a small fraction of what we get. And in general, I think per neuron, we tend to see an average of maybe like 20 active spines per 50 microns, maybe. I'd say something like that. But even branches, I mean, it's even complicated to say because individual branches vary in the number of spines that they have. It's not homogeneous. So there's portions in which there are no spines, and there's portions where the density is like 4 spines per micron. So, so I'm more or less kind of avoiding this, characterizing it directly. Yeah.
But if you had to ballpark it, what's the percentage that you see being active in two-photon?
See being active in two-photon?
Yes.
And two-photon is 25%. Okay. But then if you contribute the EM, it's much lower, right? Maybe like 5 to 10% out of the total there. Yeah.
You mentioned that you cannot see local nonlinearities with the old method, but you should be able to see it, I think, at some point where cluster of spines are active.
Yeah.
You should be able to see some local enhancement of calcium eating.
Yeah. You should.
Local NMDA spike or local calcium,
Yeah.
some nonlinearity signal, no? You
So-
mentioned backpropagation action potential.
Right. Yes. So I've spent a lot. I have looked for this. We do see features in the dendrite that may be dendritic nonlinearities. We don't see enhancement of the calcium in the spine heads when you see coactive events. And that's probably because of the biochemical isolation of the measurement itself. And it's not a reflection of voltage, it's a reflection of calcium, and there's some nonlinearities there. The one thing that is very puzzling about this measurement is that, and this is something that I have been dealing with for a while, is that the clustered activation is not very correlated with the observation of a nonlinearity of a calcium event in the dendrite. So you do see these large 10 micron hotspots that are presumably due to dendritic spikes being activated and having voltage-gated channels open in the dendrite, but those occur both in the presence and the absence of these clustered events. They don't seem to be so tightly correlated. And I can't really say why that is. I mean, I presume it has to do something weird with GCAM, but one way that we're trying, obviously, like, we want to get a better estimation of this, and so we, like many other people, are also using other indicators, like the voltage indicators, to be able to actually and I hope that we'll be able to get better estimates of what's going on at the.
There's a recent paper by Alpha
yes. I was going to show data today, but I thought it was like a little too preliminary. So we have that construct. We can express it in the same neurons in the same preparation, and we can see spine signals and dendritic signals. Now it's just time to teach the students how to do the analysis. Yeah.
So what is the role of inhibition here?
Yeah. So I mean, we don't know the role at the level of organization of inputs on dendrites and such, but we do with the technologies we have. What we see is that inhibition is more flexible depending on sort of the context of what the stimulus is and such. And so in some cases you'll see, like sort of Elon's story, like there's a co-tuning between E and I, and there's this balance. But in many other cases that we see that actually there can be, in terms of direction selectivity, actually inhibition can really quench aberrant excitation. And there is, and it comes from different locations within. There's different specific cells that are driving it in these neurons. And unfortunately, like all the techniques we have for exploring this doesn't get us down to the level of what are the exact cell types and where are they and where are they projecting to on the cell, because obviously like it's sort of a dream to be able to measure whether inhibition is on these individual branches and actually shunting of one of these branches and stuff. So that's kind of like where I hope we go. And so what we're doing now to sort of try to get along to try to measure these things is using two-photon epigenetics, getting at the subcellular level, doing some holography stuff to be able to specifically manipulate either the dendrite or individual interneurons, identify ones that are projecting to a neuron, and really like tease this apart. Because we have evidence, like I said, we have evidence that that there can be inhibition that cleaves away certain specific excitation and can highly enhance the selectivity of the soma. But we can't really say much else about it, especially at the level of we can with in terms of excitation in spine. So I'm excited. I think that's like the holy grail, because I think all the GABA sensors have not really been very helpful, and we're all these great tools are coming out and they're being very well applied to excitatory neurons, but the poor inhibitory neurons are really left aside and sad. So yeah.
I've been expecting distance from the spine to the spike initiation zone, wherever that is, to come into the picture, but I haven't heard it come into the picture.
Oh yeah, it hasn't come into the picture yet. No, I haven't. So I haven't specifically looked at that. Everything that we're doing is focused on the soma. I don't actually do much in terms of the initiations, and I think that could be
distance to the soma.
Yeah. Oh, so distance to the soma. Yeah. Okay. Yeah. So that's interesting because so one thing is all this data that I showed you today is all basal dendrites, so it's actually quite proximal. But there are some interesting relationships as you move farther away from the soma. The density of inputs gets larger, and one thing that's actually quite interesting that is can't be applied to this, but when we image, we can do functional imaging anywhere in the cell, and when we're in the apical dendrite, the statistics of the inputs in the apical dendrite look different than the basal dendrite. So actually, in fact, we see a lot more correlated activity. We see a lot more clusters up there, and that might one can suggest that maybe there's sort of that's a process that might help amplify and generate spikes that would help propagate down. In terms of the functional properties that we see here, we don't see any. There's nothing with respect to distance. There's no relationships there. Mitochondria are also more distal than they are proximal. The soma itself is chock full of mitochondria, so you don't really see anything like in the first chunk. But these, I hope, as we kind of like, these data are a little bit, it's hard. I'm always like sort of a little hesitant to really get into it deeply because the greatest distance we have is like 125 microns, which is not much, and these cells are much bigger than that. So I hope that we can, that more interesting sort of relationships emerge in the future. Yeah? Okay.
So this is layer 2-3.
This is all layer 2-3.
So, but presumably there are some inputs from FGN.
Yeah, a few.
Can you identify spines coming from FGN versus coming from?
Yeah, so Theoretically, and they're very few though. And so what's interesting is that when you just measure orientation selectivity, you would have expected to see any inputs that were like basically X or Y, so coming from X or Y, magno-particle, would be untuned, but in fact you never see an untuned input. And so this could be one of two things. It could mean that there really is very few LGN inputs, or actually the coniocellular projection is coming up. So that makes it a little more difficult. So the way that we can identify them, we can't identify them by this stimulus. But I think we can identify them by doing reverse correlation on receptive field mapping techniques and be able to more, look at the spatial, because there's well-known properties of those receptive fields, and we got to be able to map those out. And I think the other ways that people would do these things is sort of, you could also inject like opsin into the LGN and stimulate the fibers and things like that. There's obviously complications for that. I think that probably a combination of approaches, but that is something I have a great interest in, also identifying layer 4 inputs versus layer 2/3s. In the ferrets, just like the cat, the layer 4 is the primary recipient of thalamic inputs, and they're very linear cells, and then they heavily project up to layer 2/3. And they have very specific, well-characterized properties by Tony Mopshin and others, and so there's a very nice set of stimuli that I think we've sort of been playing with them to be able to be used in the calcium land of slow responses. And I think that we're actually now being able to say this is a layer 4 cell, this is a layer 2/3 cell, and this is a putative LGN cell. So that's going to be cool to see in the future too.
Thank you again.